Hi, welcome back to Cellular and Molecular Biology, or BIOL1414. Today we're going to cover Chapter 15 of Module 3 from your Biology 2E textbook. Chapter 15 covers genes and proteins. Here we have our objectives for this chapter. So first we want to be able to understand the processes of transcription and translation. Secondly, we'd like to be able to describe the processes of RNA processing in eukaryotes. And third, understand how mutation can change amino acid sequence. You'll notice that a lot of the information that we cover in Chapter 15 we've covered in previous chapters, but in Chapter 15 we're really going to dig deeper into certain vocabulary terms and processes that we haven't dug into previously. So starting off with the genetic code, remember that the genetic code refers to the DNA alphabet, those nucleotides A, T, C, and G, and the polypeptide alphabet, which would come together to make a protein, and those are 20 different amino acids. So the central dogma is that DNA encodes for RNA and RNA encodes for a protein. And I have this image on this side of the screen where you can see at the top is a strand of unwound DNA, and then we have the process of transcription, which makes mRNA for us. We'll talk about that process and kind of what has to happen before translation. And then we end up with a long polypeptide chain of amino acids that gets folded into a certain protein. So remember, central dogma, DNA encodes RNA, RNA encodes a protein. Now the genetic code is what we call degenerate because 64 triplet codons in mRNA, messenger RNA, specify only 20 amino acids and 3 nonsense codons. So in the next couple of slides, we're going to talk about a lot more of this in depth, including codons. And it says here, most amino acids have similar codons, and almost every species on the planet uses the same genetic code. So like I said previously, now we're going to kind of talk about codons and what they are, why they're important. So each amino acid, which remember we need these amino acids to form a long polypeptide chain that we can fold into a correct protein. So each amino acid is defined by a 3 nucleotide sequence. So it's a triplet code, triplet meaning 3, and those sequences are called the triplet codon. Scientists had theorized that amino acids were encoded by nucleotide triplets. So on your DNA or your mRNA, you have those little nucleotides, kind of like in the photo here at the bottom. You have all the little nucleotides here. So the DNA in this image has a blue backbone. And you're going to see T, T, C, A, C. Those are all the nucleotides we're looking at. And each little triplet of nucleotides codes for a certain amino acid. So scientists did discover that to synthesize the correct protein, amino acids have to be specified by groups of 3 nucleotides, and these nucleotides are called triplet codons. The reading frame for translation, which is the process we're starting, is set by the AUG start codon. So when the enzyme finds AUG, it's a starting point for the start codon near what we call the 5' end of the mRNA sequence. Following that start codon, which should be down here, AUG, the mRNA is read in groups of 3 until the stop codon is encountered. So there's a start and there's a stop. So basically, we clip things off at the start and begin making our piece of mRNA. And then once they hit that stop codon, they'll stop the piece of mRNA from continuing. Scientists solved the genetic code by translating synthetic messenger RNAs in vitro and sequencing the proteins they specified. So on the right side of the screen here, you're going to see this very big, long diagram about what these codons each code for. So remember, there's only about 20 of these amino acids that they could even code for. And there's all kinds of different sequences of these codons. So a lot of them code for the same amino acid. So you have to keep in mind as you're going, AUG is always going to code for MET, and that one is special because it's a start codon. But please know you do not have to memorize this for my class. I just kind of wanted to give you an indicator of, yes, we have so many different options for the codons themselves and how they're organized as nucleotides, but there's only about 20 amino acids we could ever actually come up with to make our protein. Okay, so I know that in our objectives we talked about eukaryotic transcription and understanding that process, but first I want to talk slightly about prokaryotic transcription. So prokaryotes do not have membrane-bound nuclei, which we've heard about all class. That's one of the main things that separates prokaryotes or bacteria from eukaryotes, plants and animals. Okay, therefore the processes of transcription, translation, and what we call mRNA degradation can all occur simultaneously, so happening at the same time in the same areas. Prokaryotes do use the same RNA polymerase to transcribe all of their genes. So whenever we're talking about an RNA polymerase, I have this image on the left. The big tan section is going to be that RNA polymerase. So that's what comes in after we start clipping that mRNA at the start codon. The RNA polymerase assists in reading those codons and placing the correct amino acid. So it says here a promoter is a DNA sequence onto which the transcription machinery, including RNA polymerase, binds, and then it initiates the process of transcription or transcribing that original piece of DNA. The sequence of a promoter is important because it determines whether the corresponding gene is transcribed all the time, some of the time, or very infrequently. Once a gene is transcribed, the prokaryotic polymerase needs to be instructed to dissociate from the DNA template and liberate the newly transcribed mRNA. So basically, once they've started, they have to be told to stop and disconnect from the original DNA. It says here we have something called row-independent termination with prokaryotic transcription, and it's controlled by specific sequences in the DNA template strand. And then below that, it says, as the polymerase nears the end of the gene being transcribed, it encounters a region which is rich in CG nucleotides, and mRNA folds back in on itself, and the complementary GF nucleotides bind together. So keep in mind, there's not a ton we need to remember about prokaryotic transcription other than that prokaryotes and eukaryotes perform fundamentally the same process of transcription with a few key differences. The most important being something we've heard before, which is that prokaryotes and eukaryotes transcription is due to eukaryotes' membrane-bound nucleus and organelles. So we know that those eukaryotes have the membrane-bound nucleus and organelles, while prokaryotes don't. The other important distinction is going to be that there's only one type of RNA polymerase used for prokaryotic transcription, wherein you'll find eukaryotic transcription, we have three. Okay, so now let's talk about eukaryotic transcription. And I try to keep some images here on the top right of the screen just to show you we start with the full DNA. And this is just reminding you of the central dogma we've talked about. The DNA is transcribed into RNA, which is then translated into what is going to be a protein. So eukaryotic transcription, instead of a single polymerase, so a single RNA polymerase comprising 5 subunits, which happens in prokaryotes, eukaryotes actually have 3 different polymerases that are each made up of 10 subunits or more. Each eukaryotic polymerase also requires a distinct set of transcription factors to bring the DNA template. So eukaryotic promoters, what starts this process, are much larger and more intricate than the prokaryotic promoters. And we know that to be true about eukaryotic cells in general, that most of their processes are much larger, more intricate, require more energy. So if we look at the 3 different types of polymerase, which they list in this table, you'll see that a couple of them, 1 and 3, polymerase 1 and polymerase 3, you can see what they do right here. So polymerases 1 and 3, it says the processes of bringing RNA polymerases 1 and 3 to the DNA template involve slightly less complex collections of transcription factors, but the general theme is the exact same as what happens in RNA polymerase 2. What's different is typically the product. So for 1 and 3, all of your RNAs except for what we call 5S RNA. So all of your ribosomal RNAs are going to be made by 1, RNA polymerase 1. And it says here that 5S RNA, the transfer RNAs, and all small nuclear RNAs are made by RNA polymerase 3. So this is something that you don't need to memorize. Check your study guide to see exactly what you need to know, but just understand there are 3 different RNA polymerases for eukaryotic transcription that all come out with something slightly different as a product. So then for RNA polymerase 2, this will do all of the protein-coded nuclear pre-mRNAs. So as we continue moving forward in the process of transcription, typically we're looking at RNA polymerase II doing the work for those pre-mRNAs that we're going to move forward to make a protein. And then lastly on this slide, it says eukaryotic transcription is tightly regulated and requires a variety of proteins to interact with each other and the DNA strand. Eukaryotes involve a greater metabolic investment than in prokaryotes. So basically, eukaryotes, it requires a lot more energy to fold and make these proteins. So they're very regulated and specific about what they're making as to not waste too much energy. It says here it ensures that the cell transcribes precisely the pre-mRNAs, so what we have before a completed messenger RNA that needs to be made for protein synthesis. So they're very organized, tightly regulated. They don't want to waste energy by making different proteins or different, they don't want to transcribe anything that they're not going to use. So it's trying to conserve their energy. Okay, so again, just reminding you before we move forward of that central dogma. So the reminder being that we have DNA, that double-stranded DNA, which encodes for RNA, that single-stranded, what we are going to call a messenger RNA that we're going to move forward with, that encodes for the proteins, which we know do all of the work in our body. So let's talk about those pre-messenger RNAs, because we were saying that there has to be a process that happens in eukaryotes after the mRNA begins. So there is a process of mRNA processing in eukaryotes. So after that transcription takes place, the eukaryotic, what we call pre-mRNAs, so before they can undergo the folding and protein process, they have to make some changes. So in eukaryotic, protein coding sequences are not continuous as they are in prokaryotes. So basically, there are portions that don't code for anything. And before we move forward with the messenger RNA, we like to remove those portions as to not waste a bunch of energy trying to code them. So here it says coding sequences called exons. So coding sequences meaning they contain information that can be moved forward that make a code. They're interrupted by what we call non-coding introns. So I have this image over here. This is supposed to represent a nucleus. This big light blue bubble represents a nucleus where you'll find that DNA we're talking about. And if you look closely, you'll see that in the DNA you have exons and you have introns. And in this case, the exons code for something. So they're important to move forward in the process, but these introns actually don't code for anything. And you'll see as we make that pre-messenger RNA, those introns are still there. So what happens, it says here, eukaryotic genes are composed of exons, those exons that actually code for something, which correspond to protein coding sequences. Exon signifies that they are expressed, so it's something we can move forward and express through instruction. And it says intervening sequences are called introns, and they do not have a role in coding. And it says which may be involved in gene regulation but are removed from the pre-mRNA during processing. So basically the three most important things that happen during this processing phase are listed here. So what we do is during the processing phase of the mRNA, we're going to put a cap on the 5' end of this mRNA and a tail on the 3' end. So we know where we're starting, we know where we're stopping. So those are the two important parts in the beginning. And then we also remove all of these introns. So all of these ones that don't code for anything later on, we don't want to waste energy trying to code something that we don't have code for. So those are removed. And then you have your completed piece of mRNA with those exons all linked together as a coding sequence. And you have your start cap and your stop tail so that we know when to start and stop and we're not wasting a bunch of energy running down the line. So what happens now is that this completed messenger RNA is going to move out into the cytoplasm of the cell. And it says here, transfer RNAs, tRNAs, and R-ribosomal RNAs are structural molecules that have roles in protein synthesis. However, these RNAs are not themselves translated. So the only RNAs that are going to be translated are those completed messenger RNAs that we got from our RNA polymerase II rather than 1 and 3 in that last couple of slides. So once we have that completed messenger RNA that's ready to be then coded for a protein, we move out into the cytoplasm. And in the next slide, we'll talk about where we go from here. Okay, so I'm trying to continue with the theme of all these colors to kind of, keep you on track. So remember, this little blue dot here was supposed to represent that nucleus where we did all of the pre-mRNA processing. We moved it out into the cytoplasm. So here I have it says export of mature mRNA into cytoplasm for protein synthesis or translation. So now let's talk about ribosomes and protein synthesis, also called translation. Okay. So you do not have to know all of the information listed on ribosome, tRNA, and amino acid. I do want you to know what they are and what they do during this process, but really here I wanted to explain in depth. So I'm going to kind of go into that now. It says here, after the mature mRNA, which we already matured in the nucleus and moved it out here to the cytoplasm, after it's exported to the cytoplasm, protein synthesis or translation begins in the ribosomes. The synthesis of proteins requires more of a cell's energy, or ATP, than any other process, which is why we were so specific in the last slide about cutting out all of those introns, putting a cap and a tail on the mRNA so that we know exactly what we're coding for and we're not wasting a bunch of energy doing it. So now, this big purple shape here represents your ribosome. And then this pink line on the bottom represents the backbone of that mRNA. Each of these little colors is supposed to represent a nucleotide, so an ATGC. And as I've told you guys, these guys code in triplets, right? There's a triplet codon. So what will happen is you're going to have these transfer RNAs that come in and they basically act as a reader. They come in and they read whatever triplet codon they have, and in turn they produce a specific amino acid. So how we were talking earlier about each little triplet codon codes for a certain amino acid, and some of them code for the same amino acid because there's only 20 that we get to choose from. But what happens is these tRNAs come in this process and they produce a big polypeptide, a big chain of these amino acids that are linked together kind of like beads on a necklace. And at the end, we'll move forward in a second and look at what happens. But these proteins are a long chain that gets folded to do a very specific job. So once again, that matured mRNA comes from the nucleus, leaves into the cytoplasm to be translated, and in the cytoplasm, they find the ribosome. And in the ribosome, these transfer RNAs come in to read the triplet codons from the mRNA, and then they produce amino acids in a long chain, which then are folded into proteins. So let's kind of move on to the next slide and see what protein folding looks like. Okay, so we are wrapping up the making of a protein by folding, modification, and targeting. So basically it says during and after the process of translation, individual amino acids may be chemically modified, signal sequences appended, and new protein folded into a distinct 3D structure as a result of intramolecular reactions. So I like to show on the side here what we have because I've kept the same color scheme along. So again, we're ending in that ribosome, and we remember those tRNAs had made this long chain of amino acids because they coded that nucleotide, those triplet codons from the mRNA. So it says here, every protein is made up of a sequence of amino acids that are bonded together. These amino acids will interact together to form shapes like helices and sheets, okay? And then these shapes fold up on larger scales to form the full 3D protein structure. And then what you'll see happens is the blue is the protein we've been working on this whole time, but now you see some pink added in. And that's because proteins can interact with other proteins, performing functions such as signaling and transcribing DNA. Okay, so now that we have gone through that slideshow, we should understand the processes of transcription and translation, but if you don't, they are written for you here. So in transcription, it says prokaryotes and eukaryotes perform fundamentally the same process of transcription with a few key differences. The most important difference being that prokaryotes and eukaryotes transcription is due to the eukaryotes having a membrane-bound nucleus and organelles, right? Where we know that prokaryotes do not. It says after the mature messenger RNA for translation is exported into the cytoplasm, protein synthesis begins at the ribosomes. The synthesis of proteins, again, requires more of a cell's energy or ATP than any other process, which is why those pre-mRNA portions are very important, where we remove the introns and whatnot that doesn't code for anything. And then in the third box here, we have the 3 most important steps of that pre-mRNA processing, where we add a cap to the 5' end, a cap to the 3' end, and we remove all of those introns that don't code for anything so that we're not wasting our cells' energy trying to code for areas that don't have anything. And then lastly, mentioning again that genetic mutation can alter amino acid sequence during translation, leading to improper folding and in turn improper protein production. So if we don't have the proper folding or protein production, who will do all of the work for us? Genetic mutations can cause issues like this when going through these processes. And we've talked previously about mutations in other slideshows as well. So if you want to know more information, go back a couple of slideshows and you'll learn about genetic mutation in depth. Okay, well, thank you guys. That's the rest of the things that we needed to know from Chapter 15. Please go ahead and move on and email me if you have any questions.